The aim of this procedure is to produce electrospun biodegradable scaffolds that can be sterilised and made thick enough and of appropriate mechanical strength for clinical tissue engineering. This is accomplished by first electrospinning biodegradable polymers into primary architectures, either random or aligned and orientated, or sequentially layered. The second step is to apply post-spinning modifications to the scaffolds by putting several layers together and sterilising them. Next, cells are introduced to the scaffolds and their performance during and after culture is assessed. The final step is to subject the cells to an exercise regime to mimic the physiological stress the cells will encounter in the body in order to prepare them for transplantation. Ultimately, we can assess the response of cells to exercise by measuring the extracellular matrix they produce in response to dynamic tension.
This technique is versatile and flexible, allowing a large variety of scaffolds to be made with properties specifically modified for the required application. For example, scaffolds can be designed to treat difficult conditions where tissue repair is needed, such as replacement of extensive skin loss in badly burned patients, or repair of damaged oral mucosa following cancer or trauma.
Though this video shows a lasting production of cells as a marker of dynamic stress, you could also consider a range of extracellular matrix proteins, as well as assess many other aspects of cell biology using exercised or non-exercised cells.
Generally, individuals new to this method will struggle to obtain reproducible scaffolds because the electrospinning process is affected by many variables.
Visual demonstration of the processing of electrospun scaffolds post-spinning is important. It's rarely discussed.